thank you very much for inviting me here to give a talk today, and thank you for the introduction. So I'm going to tell you about our work on the COP2 code studied by cryo-electron tomography that we've been doing over the last few years in my lab. So first of all, let me introduce the COP2 code. So as you know, in eukaryotic cells, proteins need to be transported across different membrane compartments, and this occurs by a vesicular trafficking mediated by cold protein complexes. And the COP2 code mediates transport of newly synthesized protein from the endoplasmic reticulum towards the Golgi apparatus. And as such, it needs to transport thousands of different proteins. About a third of the proteome actually needs to get into COP2 coated vesicles at the ER. So the code is essential and is highly conserved from yeast to humans. So the way that it works has been worked out through lots of biochemical studies throughout the years. The code is composed by 5 essential proteins. There's small GTPase, SAR1, and then heterodimers of Sec2324 and heterotetramers of Sec1331. And basically these proteins assemble onto the ER membrane to perform 2 main jobs. The first is to induce membrane remodeling into coated carriers that will eventually detach from the endoplasmic reticulum membrane to carry the cargos to the Golgi, and the second is to selectively bind to the cargo proteins that need to be transported out of the endoplasmic reticulum. And the process of assembly and disassembly of the coat is highly dynamic, and it's dependent on the GTP hydrolysis cycle. So SAR1 is a small GTPase. When it's bound to GDP, it's soluble in the cytosol, and upon exchange to GTP, which is triggered by an ER resident GEF, SAR1 exposes an amphipathic helix that inserts into the ER membrane, starting the membrane curvature and also activating it to recruit the other components of the coat, which eventually form this double layer coat. The inner layer is formed by the small GTPase and the Sec2324 heterodimers, which have been considered historically the adapter complex, because Sec24 binds the cargo directly or through adapters. And then the outer layer of the coat is formed by these Sec1331 rods that are assembled to form cage-like structures. So here are some of the structures that have been solved of the coat in the past decades or so. We have some beautiful cryo-EM single particle structures of in vitro assembled outer coat that show that indeed it can assemble to form these polyhedral cages, and this gave rise to a model whereby the outer coat assembly is what gives the shape and the morphology to the budding vesicle. And then we have crystal structures of the various components, which I'm not going to discuss in detail, but I'm going to use them to fit into our structure later. So what we wanted to understand is how do these coat components actually assemble onto the membrane? Because we know from various studies in cells and also in vitro that actually the COP2 coated membranes do not necessarily form such regular coated vesicles, so that these polyhedral cages cannot really explain how the coat assembles. And so in order to understand, we took an approach based on in vitro reconstitution and cryo-electron tomography. So what we do is we purify all of the COP2 components, and we use artificial membranes, in this case, that I'm going to show you today. So these are giant unilamellar vesicles. And we incubate these proteins together with a non-hydrolyzable GTP analog, and this is to stabilize the reaction before it can disassemble. And we obtain coated membranes of a variety of shapes and sizes. And then we use cryo-electron tomography to obtain 3-dimensional reconstruction of these coated membranes to analyze the architecture of the coat on these membranes. So I guess cryo-electron tomography has not really been introduced yet, probably not needed to, but essentially, in a nutshell, what we do is we put the sample in the electron microscope, we tilt it, and from the tilted images we can then obtain a 3-dimensional reconstruction. So these are just some examples of, let's see if I can play the movie. of just reconstructions, which have been like of our in vitro reconstitution samples. These have been filtered and denoised, and in particular we have used Zanetti to sort of fill some of the anisotropy that is inherent into the cryo-electron tomography reconstructions. And you can see from the raw data that indeed both on vesicles and tubules we have an outer coat layer forming these cages. Here it's visible in red. And then the inner coat layer forms a packed lattice. I mean, it's not very clear because of the anisotropy here, but you can see inner coat lattice subunits sort of making patches of lattice on the vesicles and on the tubes, which is all very good, but it doesn't really tell us at the molecular level how do these proteins interact to each other to form these different geometries, which was like the question that we were really interested in, because it will also allow us to understand how this process is regulated by various factors. So in order to understand the molecular interaction between all of the code components, we turn to subtomogram averaging. So this is a technique where you essentially can extract identical or very similar subunits out of the tomogram in silico and align and average them together. And these will give you a higher resolution structure. So I'm going to basically walk you through the structural determination of the coat that we obtain, and I'm going to start from the outside. I'm going to start from the outer coat. So I need to introduce you a little bit more about the Sec1331 proteins. So these are, just to remind you, the ones that form these cages in vitro. So essentially the heterotetramers of Sec1331 form the edge of these cages. So each heterotetramer is formed by, so the Sec31 protein is formed by an N-terminal beta propeller domain, which here you can see in red, and then a central alpha solenoid domain, which dimerizes to create the elongated rod. And then at the C-terminal half of this protein is highly disordered. There's about 300 amino acid of intrinsically disordered domain, which is highly rich in proline, and I'll come back to this. And then at the very C-terminus, there is a small domain which is predicted to be helical, but until very recently we didn't know what the function or structure of that domain was. And then in between the beta propeller and the alpha solenoid domain, there is a Sec13 forms another beta propeller that tucks in there. The N-terminal domains interact, 4 of them, to form these vertices of the cages. So we can use subtomogram averaging to analyze the structure of the outer coat on our in vitro assembled tubules, and we basically recapitulate the crystal structure and the cryo-EM structures that were obtained previously. So here we have, so we need to focus our subtomogram averaging on different regions of the reconstructed tomograms, because these are quite flexible. So if we obtain averages of the vertices, we kind of lose the information of the rods and the neighboring vertices, but by focusing our alignments on different regions, we can obtain like a pseudo molecular model of the whole outer COP2 coat. So here we have 4 beta propeller domains interacting to form a vertex, and then here we have the alpha solenoid domain. And again, here we only see the structured regions, we're not seeing the disordered region. But one interesting thing that we noticed here that was not seen before in the crystal structure is that if we kind of play with the threshold, we can see that there is a very well-defined domain that appears on the rod. And because we are not adding any other protein here apart from the COP2 proteins in our reaction, we hypothesized that that might actually be the C-terminal domain of Sec31 that I mentioned before. And just to cut a long story short, we confirmed that it is. So first of all, if we create a model, whether it's homology modeling or AlphaFold, it fits quite well into this extra density. And I have to say that AlphaFold was quite impressive because it also predicts the interaction exactly at the right spot. And also we did mutations where we truncated the C-terminal domain, and we used the truncated protein to reconstitute budding in vitro. And what we observed is that, first of all, the protein is functional. It does induce budding in vitro. And we obtained tubules, so I guess the contrast here, the resolution is not great, but you have to trust me that there is outer coat here visible in the tomogram, but is actually not as well ordered as for the wild-type protein. But nevertheless, we managed to do some low-resolution subtomogram averages from this disordered outer coat, and we noticed that that extra blob is missing indeed from the averages. Here you can see pointing to the orange arrows, confirming that that's the C-terminal domain. So essentially, what we are seeing at the level of the outer coat is that not the network that forms these cages is not only mediated by the N-terminal vertices, but there's also the C-terminal domains kind of binding onto the rods. And we don't know what the role of that is, because they could be binding in cis onto the same tetramer, but there's no reason to think that they might not bind in trans, actually reinforcing this network of interaction. And moreover, something that I haven't mentioned yet, the disordered region that sits in between the N-terminal half and the C-terminal domain is the one that goes and reaches down and binds to the inner layer of the coat. So this interaction probably reinforces also that kind of inner to outer coat interaction. So to understand the role of these two nodes of the network, we decided to do mutations. So first of all, we started off with an N-terminal truncation mutant, which we actually designed as a control because we didn't think that it would be active at all. But then when we actually did the Badin reconstitution, we were very surprised to see that this was active in formation of tubules. We didn't see any vesicles, only tubules. But they were clearly there, and the inner coat was ordered. This is just a negative stain image, so you're going to have to believe me, but the inner coat is ordered there. But then we kind of turned to our collaborator at the LNB, Liz Miller, to do some genetic assays on this, and she found that the yeast without the N-terminal domain actually are completely dead. So the N-terminal domain is not essential in vitro, but is essential in vivo. And a kind of a very nice experiment that Liz has done was to repeat this genetics assay using an yeast strain that is lacking a protein called EM24. And this is a receptor for GPI-anchored proteins at the ER. So this helps GPI-anchored protein getting recruited at COP2 exit sites, meaning that these yeasts have ER membranes that are devoid of GPI-anchored protein clustering at COP2 Badin sites, making the membranes a lot more deformable. And in these yeasts, then the N-terminal domain is not essential anymore. So we actually repeated the same set of experiments with the C-terminal domain, and we essentially saw exactly the same thing. So the budding works, I showed you in the previous slide, and the genetics results were exactly the same. So it is an essential domain, but it's not essential to promote budding per se, it's essential to promote budding of rigid protein-rich membranes. Another thing that we noticed when analyzing the structure of the outer coat was that there were some rods that were sort of cutting in between these lozenges of the cages, which were not seen before in any of the previous studies. And we were kind of wondering, what that could possibly mean. And by doing some kind of data analysis that I'll spare you the detail of, we found that actually some rods were having a green interaction, so like between the N-terminal beta propeller domain and the middle of another rod on one side, and on the other side, a vertex, an N-terminal domain was interacting as part of a vertex. And one of the advantages in tomography is that you don't only get the structure, you can also place your structure back into the tomogram and analyze its geometrical relationship with the neighboring structures and neighboring proteins. So when we do that with all the various bits and pieces of outer coats that we saw, we found that most of these sort of hybrid rods were sitting so that they kind of glued together patches of outer coat lattice that were not quite in frame with each other. So we think that this type of interaction, of novel interaction we see, might actually help to stabilize the outer coat lattice on membranes that have continuous changes in their curvature. Right. So what about the inner coat? So again, we apply subtomogram averaging at the inner coat level, and this is actually a much nicer sample, and it goes to much higher resolution. So I want to show you the latest one that we obtained, where we improved the resolution significantly thanks to actually moving to the Lion 4. So this is quite recent work that we've been doing. So basically, the inner coat I showed you before, there's heterotrimers of SEC23, SEC24, and SAR1. So the SAR1, as a reminder, is the small GTPase. SEC24 is the main cargo binding subunits. And SEC23 has multiple roles. On one hand is an adapter protein that binds all of these proteins together. And the binding site for the outer coat, the flexible domain that I showed you earlier, is actually on various pieces of SEC23, I'll show you later. And SEC23 also has another function, which is to stimulate the GTPase activity of SAR1, which otherwise it's a pretty poor GTPase on its own. So what can we learn from, so this structure is, we obtained, as I said, with Reliant 4, and I just want to kind of briefly mention some work that Ewan Pyle, a postdoc in my lab, has been doing to, those of you who have been doing cryotomography know that very often you have to use a lot of ad hoc in-house scripts to kind of go from one place to the next. There's even though pipelines are being developed, this is happening now, and so Ewan has developed a pipeline that basically takes, in a couple of commands, takes images off the microscope and basically makes everything ready for relying for subtomogram averaging. So if you're interested in that, it's all on GitHub, and I'm sure Ewan will be very happy to help, and also thanks to Alistair Burt for a lot of input in this. So that's the end of the plugin. And so here is the new structure that we obtained, and I'm just kind of playing this movie. So I don't want to give numbers for the resolution. I don't really trust them. It's somewhere between 3.5 and 4 angstrom, I think. But I'm just showing you this to show that the quality of the density is really, really good. We see every single side chain for the vast majority of the protein, and they're very, very well defined. So we're very happy with this result. And if I manage to move on to the next slide. But what can we learn? So we can look at a number of different things. For example, let's start from the interaction between the inner coat and the membrane. So one of the advantages of looking at these structures in situ, is that we can also analyze how they interact with the membrane. So for example, I mentioned that the primary interaction of the coat with the membrane is of a SAR1 amphipathic helix, which gets exposed upon GTP binding and inserts into the membrane. And we can see very clearly from our reconstructions that this amphipathic helix actually bends about 90 degrees and sort of horizontally embeds in the outer leaflet of the membrane, and this is what helps the membrane achieve its curvature. So here you can see just a slice through, and here is a 3D rendering. All of the crystal structures of SAR1 are missing the amphipathic helix, so, but we can clearly see that's where it gets truncated. And funnily enough, Rosetta predicts this 90-degree kink quite well, but AlphaFold doesn't. AlphaFold gives you just a totally straight helix there, which is quite interesting. Maybe a proof that we need more membrane protein structures there. Other observation about the interaction with the membrane that we can do with a higher resolution structure that we couldn't see before. So we know that there is a requirement for negatively charged phospholipids for the coat to actually bind and induce tubulation, vesiculation of the membrane. And we see, for example, in some places, this is the nicest one, clearest one, there's lots of water coming down, but is that compared to the AlphaFold 2 or crystal structures, there is a loop here carrying a histidine which really gets pulled down towards the membrane, probably by an interaction with some negatively charged phospholipid head. We can also tell about the interactions between the inner and the outer coat, even though they are mediated by a disordered domain. And here again, there's not too much scope for actually traditional structural studies on disordered domain, of course. but some parts of this domain are bound to the inner coat, and we can visualize them. So, as I said, this domain is proline rich, and it contains several triple proline motifs which have been shown before to mediate interactions to the Sec23 molecule on the inner coat. It also contains a GTPase activating protein. It also contains a peptide that increases the gap activity of Sec23 by 10-fold. And it also is highly positively charged. So that's sort of, okay, so how can we visualize where all these bits and pieces are binding? So we can, of course, because the binding is probably substoichiometric and it's probably quite flexible because it's a highly disordered and long domain where there's several triple proline motifs that are all different from each other. These regions do not show in the high resolution structures, but we can visualize them if we apply a positive B factor to our final reconstruction. We can see the blobs of the lower resolution parts. And so these colored in red are all the blobs that are not explained by the molecular model of the inner coat. So they must be accounted for by something else that is binding to the inner coat. So, and basically, I just want to kind of be quick here, but what we are seeing is the triple proline motifs are actually, we know they're binding here because of the previous crystal structure that shows triple proline binding here. And what we are seeing is that actually we don't only see the region around the triple proline, we see a much more extended region sort of gluing between neighboring inner coat subunits here, and I'll come back to that. Then we see these little blobs here, and we kind of notice them because that's where the GTPase activating peptide binds according to crystallography. So we only see very scattered density there, so probably this binding is not very stable. And we also see a very prominent blob here, which when we look at the surface representation of the inner coat, we see that all of this region here sits on a highly acidic surface. And knowing that the whole 300 amino acid of disordered domain are very positively charged, we think that these basically non-specific charged interactions that are going on there. And then we also see this other blob, which we can't really explain with interactions with the outer coat, and what we think this is Sec20. So this, sorry, I should have put another. So this, that blob is there, and here is another Sec24 subunit. So there's another row of the lattice up there, and we think that that's an interaction site of Sec24 because it's a Sec24 crystal contact from previous work. So anyway, we can map a lot of these interactions at a medium to high resolution. We can also look at the interfaces between neighboring inner coat subunits. So here we have a Sec23 from one subunit with the SAR1 GTPase in between, and another Sec23 subunit there. And the interface is quite extended. I've already mentioned the triple proline motif that sort of extends to glue in between neighboring subunits. We also see many other interactions, and one that really caught our attention is this loop that I have here in cyan, and we modeled as a helix, that is totally disordered in all of the crystal structures from all species that have been solved, but it clearly becomes ordered in the context of the assembled lattice, and it clearly makes an interaction with the neighboring SAR1. So we were quite intrigued by that loop, and we decided to make some mutants. So we removed that loop, we substituted with a shorter stretch of glycine and serines, and we were quite disappointed to see that this protein was perfectly functional. It made a lot of nice tubules where the inner coat lattice was perfectly ordered. But then we thought maybe there are other places in the interface that can be sufficient to mediate the lattice formation. So we combined this mutation with a mutation on the outer coat where we depleted all the 7 triple proline motifs with the aim of also removing that sort of gluing activity that I showed you before. And we had a very striking result where we couldn't see any tubules, we only saw round vesicles. So what we kind of concluded from this is that if you don't allow for GTP hydrolysis by using non-hydrolyzable analogs like this in our system, the inner coat tends to assemble and form tubular lattices. When you disrupt that interaction when destabilize that interaction, then the outer coat, which tends to form spherical cages, wins the kind of tug of war, and you have more spherical cages. So this suggests to us that regulation of the GTP hydrolysis cycle and of the assembly-disassembly dynamics of the coat is very important to regulate the morphology of the budding vesicles. And that is probably very important because there are various factors that are known to interact with the COP2 coat, and they are known to affect the GTP hydrolysis rates, and these factors are essential for transport of, for example, very large cargoes out of the yard, for example, like procollagens. So there's clearly scope for trying to understand how these factors work and how they affect these assembly structures. Okay, so I'm finished. This is my summary slide. I showed you that we can use in vitro reconstitution and cryotomography subtomogram averaging to really kind of resolve at high resolution enough for molecular interpretation various regions of the coat as it is assembled on the membrane. We identified numerous interactions that form this kind of network, and this allowed us also to go and mutate various points in this network to understand what the role of all of these interactions is. So I'll finish by thanking my group, especially work today from Ewan and also Josh, who's now left, moved on to San Diego, and all the people that have been involved, past and present, especially collaborators Liz Miller at the LMB, who did the genetics I showed you today, and many other assays that I didn't have time to go into. Alistair and Shores for the Reliant 4 sort of kind of developments. The data was actually collected through INEX at EMBS, so big thanks to the INEX team and to Bim and Felix and Jean-Guy for data collection, and everybody in the department for lab and IT support and funding. And thank you for listening.